In this video, I'm going to go over the neuron and how electricity travels down the neuron, how it conducts electricity. Remember when you learned about nervous tissue, its function was conduct. So let's just go over, and you probably already have gone through this though, the parts of a neuron. So you have the dendrites here, which are going to collect information through neurotransmitters from other neurons or organs of the body or so on. So again, they'll take information using neurotransmitters, and then depending on what kind of information they're getting, it can send an electrical signal down the neuron through the cell body. And then here you have the long axon, and then here you have the axon terminals. And if you remember, we talked about synapses, so these contain neurotransmitters, and then depending on the kind of signal that's coming down the neuron, it will release neurotransmitters into the synapse and stimulate an organ, stimulate a muscle, stimulate another neuron. Or it can actually inhibit another neuron or stop or decrease the firing of another neuron. So that's the basic anatomy. Remember the myelin sheath. I'm going to show you today how that's going to play a role in the propagation of an action potential. Okay, so one of the things you have to understand that we haven't spent a lot of time on that's kind of unique here is the ion channels that are located within the cell membrane or on the cell membrane of neurons. So if you could look at this at a microscopic level, you would see all these ion channels. Now you've heard of channel proteins. We did those, I think, in test 2. You know what ions are. So these ion channels will let ions transport back and forth between the inside and outside of the neuron. Now, what does gated mean? Gated means that they have doors that they can open and they can close. So there are going to be times when these gates can be closed and ions can't move, and there's going to be other times that the gates are open and the ions can move. So that's what gated means. Think of them as a door. Now, what is the difference between a chemically gated channel and a voltage gated channel? The difference is what causes the door to open. So with chemically gated ion channels, a chemical, specifically in this case a neurotransmitter binding to the channel, will cause the door to open. So when the neurotransmitter is not there, then the door is closed and the ions can't move. So some neurotransmitters are going to open sodium gated channels. Other neurotransmitters are going to open potassium gated channels. But again, if that neuron is not there, if the neurotransmitter is not there, it's not going to open. These chemically gated channels are located in the cell body and the dendrites of the neuron. So remember, other neurons are sending signals through neurotransmitters. Certain neurotransmitters are going to bind, some are going to open sodium gated channels, others are going to open potassium gated channels. The second type of gated ion channels are voltage gated channels. These respond to the voltage of the neuron. Voltage we're going to talk about as a charge. So when a neuron is at a certain charge, certain voltage gated ion channels are going to be closed. If the voltage changes to a specific another different voltage, then the door is going to open. So voltage gated ion channels will open and close in response to different voltages or different charges of that neuron. Those are going to be located, and these are the ones we're going to focus on. These are going to be located all along the axon and the axon terminals of the neuron. So chemically gated channels are here and respond to neurotransmitters. The voltage gated channels are going to be located in this region of the neuron. Okay, I mentioned sodium potassium. Those are the two ions we are going to focus on in terms of being able to conduct electricity down a neuron. All right, so a brief definition of what voltage is. Voltage is potential energy. The separation of charges. The best example I can use with you is a battery. So we have a positive end of the battery and a negative end. That means we have voltage, we have potential energy, and there's something in here in the battery that is separating those two charges. So it's the potential between 2 points. You can either measure it in volts or millivolts. We're going to be talking about millivolts because we're being talked about little bitty things, okay, little voltage. So your neurons work like a battery does. If you just hold a battery up, there is potential energy there. You've heard of a 9-volt battery that has voltage, it has potential. But in order to get that electricity to flow, you're going to have to connect the ends. So we're going to have a positive side of the cell membrane, a negative side of the cell membrane. The cell membrane is going to be what splits the charges plus and minus. When you have voltage gated channels, they're going to be shut. So you got a positive and negative side, positive on the outside, negative on the inside of the neuron. When those ion channels open, now you're going to see ions that are going to flow. That's going to generate electricity. Okay, that's going to generate electricity, which right now I know that probably doesn't make sense, but it will as we go along. Okay, so when we look at a neuron, if we take a little voltage meter and we stick it inside the axon, we will find that the inside is negative compared to the outside, specifically around negative 70 millivolts. So the inside is negative compared to the outside, which is positive. Now, why is that? One reason is because these things we're going to talk about here is how the ions are set up in a resting neuron. So I'm going to do it in words and then I'm going to show you a picture. So inside the cell, the potassium is very high compared to the outside where the potassium is low. Okay, so what we've done is we set up a concentration gradient. Inside, very high in potassium, outside, very low. Now you might be thinking to yourself, well, potassium, we talked about diffusion. Why doesn't the potassium go from high to low? It will, but in a resting neuron, it cannot because the potassium ion channels, the voltage ion channels are closed. So it keeps the potassium from moving. On the outside, when we look at sodium, sodium is very high on the outside of the neuron and low on the inside. And again, the reason it's going to be high on the one side and low on the other, why doesn't it move? Why doesn't sodium go from high to low? Because again, these voltage gated channels, when closed at rest, it keeps the sodium from moving. So that means the inside is negative compared to the outside. Now you may be thinking to yourself, hopefully, well, they should be zero because the potassium and the sodium are going to equal each other out. So let's look at this picture. All right, this is one of my lovely little drawings here that I did. This is what a resting neuron looks like. Okay, so you'll notice on the outside, the potassium is high compared to the inside where it's low. Again, why does that stay that way? Why doesn't the sodium just go in? Because we have these voltage gated channels. Okay, so the sodium is red, and so you see these red voltage channels, they're closed, so the sodium cannot move. Okay. Potassium, very high on the inside, low on the outside. Again, it wants to move, but at rest, these potassium voltage gated channels are closed. Now, I told you that if we stuck a voltage meter in here, the inside would be negative compared to the outside. When you look at this picture, that shouldn't make sense because it looks like the charges balance each other out. So the reason that it is negative compared, the inside is negative compared to the outside, is because of these structures called the sodium-potassium pump. Now I only have one on here, but they go all along the cell membrane of neurons. And what these pumps do, they are pumps, they are going to pump against the gradient. So here's what the pump does. It pumps 3 sodium out for every 2 potassium in. Now, look at it. Is this pump going with or against the gradient? Is it going with or against the gradient? If you look at this, it is pumping against the gradient. It is pumping from low to high, low sodium to high sodium. So it's having to push sodium against the gradient. Same thing with potassium. I know this is hard to see, but this is 2 potassium. It is pumping against the gradient. It's pumping 2 potassium from low to high. So when you're working against the gradient, when the body is pushing against the gradient, what does it need in order to do this? Go back in your memory bank. What it needs energy in the form of ATP. Okay, it needs energy in the form of ATP. So these pumps are constantly working against the gradient. So that's the one thing you need to know. You need to know what it pumps, in which direction, that it needs ATP because it's going against the gradient, and that this sodium-potassium pump is the reason the outside is positive compared to the inside. And here, let me explain that. So we're pumping out 3 positives, but we're only pumping in 2 positives. So in comparison to each other, right, in comparison to each other, the outside is more positive than the inside. So the outside is more positive because we're pumping out more positives than we're pumping in. The outside is positive compared to the inside. So when we stick a voltage meter in this neuron, it's going to have a negative potential, specifically -70. Okay? So if I ask you, questions I could ask you, where's the sodium high? Where's the sodium low? Where's the potassium high? Where's the potassium low? How do we get the negative voltage in a resting neuron? And you would talk about this. Okay. You may need to go back and listen to this section again. Do that if you need to. Okay. Now what we're going to talk about is how do we conduct electricity. So I told you, just like a battery, we've got a positive side and negative side, and we've got the cell membrane and these closed ion channels that are keeping that potential. And then remember what I told you is when you connect the 2 ends of the battery, then you'll get an electrical flow. The way we're going to connect the 2 sides of the battery in this case is we're going to open these ion channels. They're voltage gated channels. So as the voltage changes, these are going to open and close. Okay, so now I'm going to go to this picture, okay, and you probably might maybe see a picture like this on the test. This is showing an action potential. What an action potential is the signal starting on one end of the axon and going all the way down the axon, conducting electricity all the way down the axon. Okay, so here we are at resting. That resting voltage is caused by the sodium potassium pump and the fact that your voltage gated channels are closed. What happens is, I'm going to scoot back here. Okay. So what's going to be happening down at this end, which we're not spending a ton of time on in this class, you will if you go on to a 2-semester class, is neurotransmitters are binding. And we may have excitatory neurotransmitters which are going to try to excite this neuron and induce an action potential. They do that, those neurotransmitters do that through those chemically gated channels and making the neuron more positive. There are also inhibitory neurotransmitters. When they bind, they can actually make the neuron more negative and then it won't fire. What we are going to focus on is when it fires, when it induces an action potential. So imagine a bunch of neurotransmitters coming up here and changing the voltage at this end of the neuron. Okay, so what will happen is when we have excitatory neurotransmitters binding, we're going to go from -70, we're going to get to -50, and that is considered threshold. Okay, once threshold is hit, then what's going to happen is the electrical signal is going to conduct all the way down the axon. Once it starts, it's not going to stop. All right, so what's going to happen in that case? What is going to happen is at -50, at threshold, the sodium gated voltage channels open. Okay, the sodium gated voltage channels open. So imagine these red gates opening. What is sodium going to do when that happens? Sodium is going to go from a high concentration to a low concentration. It's going to rush into the neuron. It's going to rush into the neuron when we hit threshold and the sodium gated ion channels open. What is that going to do to the charge of the neuron? That is going to make it more positive. Okay, that's going to make it more positive. And that is called depolarization. So from here to here is depolarization when the sodium gated channels are open and sodium is rushing into the neuron. Now something happens when we get up here to this +40. What's going to happen is the sodium gated channels are going to close and the potassium gated channels are going to open. So we get to positive 40, these close and these open. And what's potassium going to do? It's going to rush from a high concentration to a low concentration, and now you have all these positives leaving the neuron. What is that going to do to the charge of the neuron? It's going to make it become more negative or go down. That is called repolarization. That is when the potassium gated channels are open and potassium is rushing out of the cell. Okay, so that is an action potential. Now what happens though, it propagates down the neuron. Okay, it propagates down the neuron. Okay, so this just, look, if you look at this, this is just showing you repolarization, depolarization. Repolarization, remember, sorry, depolarization, sodium is rushing in, then those sodium gated channels close, potassium opens, and then the potassium rushes out. What it's going to do, it's going to propagate down the neuron. Okay, it's going to propagate down the neuron. So it's going to look like this. It's going to start right here. This neuron hits threshold. Once it hits threshold, that means the signal is going to go all the way down to the end of the neuron. It's not going to slow down. It's going to go through a continuous speed all the way to the end. So it propagates, it moves. Sodium channels open here. Then what happens is they close, potassium opens, but then sodium opens here, potassium opens here. Sodium, potassium, boom, boom, boom. These ion channels are opening and closing all the way down the neuron, okay, at a speed faster than we can even imagine. Okay, at a speed faster than we can even imagine. So you've got to constantly kind of view or think about sodium opening here, sodium rushes in, they close, potassium opens, potassium rushes out. As that's happening, it's being propagated down the rest of the neuron until it gets to the end. That is an action potential. Okay, that is an action potential. Now, this neuron can fire very quickly, and it may have to fire again very quickly. So what happens? How does it get back to its resting potential? How do we get the sodium back where it was and the potassium back where it's supposed to be in order to get back to resting so that it can fire another action potential? What it's going to use is that sodium potassium pump. Okay? So as this is propagating down the neuron, and the sodium potassium are all in the wrong spot, what's going to happen is these pumps are going to pump them back where they need to be so that this neuron could fire another action potential very, very quickly within milliseconds. So it fires very quickly and then it goes back to resting very quickly and can fire again. All right, and again, what gets it back to the resting potential is this sodium potassium pump, which remember needs ATP. Okay, needs ATP. Okay, so the all or none law, which I've already kind of mentioned, I just didn't call it this, basically says that once threshold is met, once we get to that negative 50, then the action potential is going to be generated and it's going to go. It's going to fire down the axon, down to the axon terminals, and it's not going to slow down. It's going to go all the way to the end. So you can, the way to control a neuron is down at this end. You can either make it not fire or fire more, because once it hits threshold, it's going to go. Okay, it's going to go. All right, so what role does myelin sheath play in this process? Here's an example here showing you the myelin sheath, and you remember the nodes of Ranvier in between the myelin sheath. Myelin sheath is going to speed up the action potential, because what can happen is without myelin sheath, okay, without myelin sheath, the action potential has to go through every sodium potassium channel all the way down the neuron. Okay. With myelin sheath, the action potential can jump from node to node. Okay. So it's going to go much faster. Without it, it has to go down the neuron every channel opening and closing as we go down. With myelin, it can jump and it can get down to the end very quickly. Okay, very, very quickly. So without myelin, it's called continuous conduction because it has to continually go down the whole axon. With myelin, it's called saltatory because it can jump from node to node. So in your body, when a signal is really important, you're going to find it has neurons, okay? It has neurons that are myelinated. If the signal can travel a little bit slower, then they're not going to have myelin. The other thing that affects the speed of a action potential is going to be the diameter of the axon. When you have a larger diameter, it's going to travel faster. Okay, it's going to travel faster than a smaller diameter neuron, okay, axon. So again, your body, where signals have to go faster, you're going to have larger diameter myelinated fibers. Okay, if you've ever, the best example, easiest example I can give you is if you're watching a basketball game, let's just use basketball as an example, and someone comes down and their ankle twists, okay, and you always have to watch those things, they show it over and over again on TV, and they're just writhing in pain. They're writhing in pain, and you're like, my gosh, they broke something. So they're writhing in pain, they come out and get them, and then they put them on the bench, put some tape, blah, 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 and then 5 minutes later, sometimes they're back in the game. Why is that? Because when your body is doing something or your body is put in a position that it should not be, your neurons, your sensory neurons, want to get that signal to your brain very quickly that we have been put in a situation we do not like. So as that ankle rolls, there's going to be pain signals that are going to be sent up sensory neurons. Those neurons are myelinated and they have a large diameter because the signal we want it to get it to your brain as quickly as possible so you stop twisting it. Okay. Now, after you get past that writhing pain and you sit down, as long as you didn't do any major damage, you'll have this throbbing pain, okay? It's not as severe. That throbbing pain is being sent, hey, we've done some damage down here. It's going to be sent up neurons that are smaller in diameter that might not be myelinated because it's not as important to get to your brain as quickly. Okay. So that's just an example. In situations where people lose myelin sheath, and there are diseases like that, it can significantly shorten their lifespan because they won't be able to send signals where they need to quickly enough. Okay, this is not an easy topic. This is not an easy topic, and this is just showing you the myelin. Um, it's perfectly normal for you to have to go back and listen to it again and take some notes on it. This is one of those that it's not expected that you get it the first time. You may hear it in class and then you want to watch this video. Or you may have to watch the video 2 or 3 times, and that's perfectly fine. Just go through it, take notes, and then come up with questions you might have and make sure you get those answered before the test. All right, talk to you soon.